Fine. Sorry, we missed your whole beautiful intro.
It's okay. That is perfectly fine. And I think we probably still have, I'm not sure how many participants were you expecting?
I think probably about this number. Let me see how many RSVP. So 10, yes. So, and we are at 9. So that's pretty good.
Okay.
Yeah. Yes.
Okay, awesome.
Maybe go ahead and start, and I can. Well,
Sure.
I have the system on, but I didn't load the sample or start the software.
Okay, sure. So we'll start with first just an overview of the system and some of the physical aspects of it. We don't need the software quite yet. Maybe what I would ask is if you can maybe grab the webcam and maybe point it at different locations on the CD7 as I walk through the physical interaction parts of it. I'm going to stop my own video sharing so that way we can focus just on your own screen. And I think everyone can sort of adjust the screen size, right, to see the video from Holly front and center.
I don't have too much range on this camera, but
that's okay.
Get the idea.
Yes. Okay. Yeah, that's perfect. So I'll start with just a brief description here of the CD7 as we have this view. So as you see, the CellDiscoverer7 is a fully enclosed system. That's because everything inside is fully motorized and automated. The idea is that the CD7 can do a lot of the prep that you would do on a traditional microscope. You can do it for you. That way you can make the setup for your acquisition more easily. This particular CellDiscoverer7 is fully equipped with both Widefield and Confocal capabilities. So inside the CD7, we actually have an LSM900 with the Airyscan 2 detector as well. That way, this system really has the best of both worlds and can capture both high resolution images and also benefit from automation features in the same system. As you see here, the system can accommodate different kinds of samples. So we have here a few sample holders for 35mm dishes, 60mm dishes, those non-8-well chambers slides. We can obviously also accommodate standard slides. In addition to all of these formats, we can also accommodate well plates. So your tissue culture well plates would fit exactly right in the loading dock without the need of any adapter. And the CD7 can image perfectly through both plastic and glass bottom well plates. So if you have a standard tissue culture plate, imagine like your 12 well, your 24 well, your 6 well plate that you maybe are just using for cell culture, you can definitely do that on the CD7. And the CD7 will actually be able to detect the kind of sample that you're putting in. I'll talk a little bit more about what that system does. As you can see here, this is how you would load a plate into the CD7. And you do have that little mark on the top left that will indicate to you that well A1 should be on the top left. And when you place a plate in the CD7, it really only fits one way, but maybe you might want to make sure that the plate is sitting flush and flat on the bottom of the loading dock. There's a little bit of wiggle room, but not too much. The plate should definitely fit in completely flat without any tilt. Maybe if you misplace it, then you'll notice that the plate will be tilted. So it's a good idea to just double check, make sure that everything is nicely in place in here. So that would be sort of a brief overview on the different type of samples that the system can accommodate. Like I mentioned, the system can do imaging through both plastic and glass bottom plates, and I'll tell you a little bit more about how that gets adapted internally. Before we dive into that part, though, I'll keep on by just mentioning a few of the other features of the CD7. The CD7 does have full incubation control for both CO2 and temperature, as well as humidity. So it actually has a built-in humidifier so that if you want to do a live time-lapse experiment, you can definitely do so on the CD7. I've seen everything from, just a couple of hours all the way to a few days or even a couple of weeks. So the CD7 has very stable incubation because it's fully enclosed. That makes it possible for you to do those long-term time-lapse experiments and make sure that your cells are going to be happy in the system. It essentially acts as its own incubator. We also see here our automatic water immersion. So in the CD7, you do have a high resolution objective that goes all the way to 100x. And it does work with an automatic water immersion system. So you don't have to deal with the immersion yourself. Whenever you need this objective, the CD7 will call that objective into place and will apply just the right amount of water immersion when needed. And when you're done with it, it'll also suction off that immersion and place it in that waste container. That way, if you need to go back to a lower magnification objective, you can do so, and you don't even have to take your plate out. Okay, there's a good chance that you won't even need to open this, this door at all. The humidifier and the immersion does last for a long, long time. Okay, you'll also see that above that door, you have the LCD screen, and it is a touch screen. So it is actually possible to even build experimental workflows that you can select from a drop-down menu on the LCD screen if you wish. This LCD screen also will act as a status update screen. So when you're in the middle of a workflow, it'll actually say what step is it doing, if it's doing a Z-stack, or whatever it is, to give you a little bar with the remaining time left.
Yeah, so sorry, my camera cord isn't long enough to show the touchscreen very well.
It's okay, that was perfectly great.
Okay.
Okay, so that's a very brief overview of the system. I guess I'd also mention, if you are doing live cell experiments and you'll need CO2 control, you will need to make sure that you do open the gas tank that's all the way to the end at the entrance of the room, just to make sure that the gas tank is ready to provide airflow to the CD7. Okay. Now I would like to go through how you would do a full power cycle on the system. So power on and power off procedures. And I'll describe the steps for that. So right now the system is on, but the software is closed, right?
Yes. Oh, yes.
Okay. So I'll describe first how you would do a full power off since the system is on. The first thing that you would do when doing a power off is you do want to make sure that you first close the Zen software. And there is a little microscope icon in the taskbar that I'll point out when we open the software. But that little symbol signifies the connection in between the computer and the microscope. So you would first close the software, close Zen, then wait for that service tool to disappear, and then you can turn off the microscope. To do this, you would just press the circular button next to the LCD screen. So there's a little round button right there. And that would essentially put the CD7 in a sleep mode or standby mode. If you're maybe the last user to use the system for the day, you could just do this step and just leave the system as it is. It is in sleep mode, and that's perfectly fine from one day to the next. If you do need to do a full power cycle, then we would actually then fully power off the system by flipping the hard switch off, which is right there on the bottom of the system. You see that switch right here. And then after turning that switch off, then you would also turn off the power strip that's right on top of the PC unit. And then that would be a full power off of the system, full power cycle, which you would want to do maybe if it was Friday night and nobody was going to use the system over the weekend. Or sometimes I have seen that maybe there is an error that you're seeing come up on the software. There's a good chance that that error is a miscommunication between the microscope and the computer, and usually a full power cycle, including turning the hard switch and the power strip, will get rid of this error. Once you wait a minute or so for the full system to power off, then you can power the system back on again, and it's essentially everything in reverse. So first you would do the power strip, then the hard switch on the front bottom of the system. And then you would press the circular button next to the LCD screen. Now, when you do that, the LCD screen will turn on and it will give you a brief update, and the system will take a minute or two to initialize. It will actually give you a status bar of where it is in the process, but you do want to wait for the system to fully initialize. You see the bar right there before opening the software. And this is very important because, again, the system is fully automated and it communicates with the computer. So it really has to fully initialize before starting that connection with the computer. So make sure that you do wait for that full status bar to be completed before opening the software, which will be Zen 3.4, and it will be located on the desktop for the workstation. Okay, any questions about power on, power off, or any other of the physical aspects of the system so far? Okay, very simple, right? Like you see, there's no eyepieces, there's no extra buttons, there's no knobs, there's no switches. The CD7 is very simple on the outside. It was designed to be more user-friendly and to require less user input. And again, that's thanks to the interaction of the system controlled by the workstation. Okay. One other thing that I'll mention while the system finishes opening or initializing is with respect to the samples. Again, like I mentioned, you can feel free to bring in your traditional tissue culture plates, for example, a 6-well tissue culture plate. Those are typically plastic plates and they're fairly thick. Or you can have your glass bottom plates. It could be anything from a 6-well to a 12 to a 24, 48, 96, 384, whatever standard plate you may have. It'll work with the CD7 as well as the other formats that I mentioned earlier, 35mm dishes, 60mm dishes, non-coil chamber plates, BD slides, regular slides. The system is actually very flexible in the type of samples that it can accommodate. Also, if you're doing a long-term live experiment and you are worried about possible contamination, the CD7 actually comes with a UV disinfection unit. So if this is your goal, you may want to run a UV disinfection cycle of the inside of the chamber. That way the inside of the chamber is sterile, and it'll add another layer of protection to prevent contamination for your sample. Okay, there you go. The system finished initializing. So at this point, it's okay to open the software, which you do with the icon right in the middle of the desktop, that Zen 3.4 icon.
Here's the detecting light.
Right, and that is the UV decontamination unit. It's essentially the size of a well plate, so it can be loaded directly into the loading dock. And it even has some guides on one side. This is like electrical connection on one side, and it'll have marks on the loading dock as well as to where that goes. And when that is loaded in, the system will actually recognize it and we'll ask you, do you want to run a disinfection cycle? Okay. Let's go ahead and open the software and let the software initialize as well. You'll see when you open the software, you have a couple of different options on how to run it. You want to select the option that is the CellDiscoverer option. If you want to communicate with the microscope, there is also an option that's just for image processing, in which case the microscope can be off and you're only doing image processing. But for the most part, you're going to be using the actual CellDiscoverer connection. So that would be that option right there. The software will also take a few seconds to initialize as it initializes the hardware and starts that connection with the CellDiscoverer7. I'll also point out here, now that we've opened the software, you'll see that service tool icon that I was talking about, that little microscope icon on the bottom of the toolbar. And maybe, let me see, where am I here in the connection? It seems, Holly, it seems that my TeamViewer connection only has one monitor. Do I, oh, okay, there we go. I just selected the other monitor. Now I can see this. Oh, perfect. Yes, so you were circling that microscope icon there. Thank you. So that's where it is. And you'll see that that represents the active connection between the computer and the CD7. So if you were doing a power cycle, you want to make sure that you first close in and then wait for that service icon to go away, that send service icon, before actually turning off the CD7. Okay. Let me minimize some of these options here. All right, so that really is it when it comes to the physical interaction with the system. The one other thing that we'll do is go ahead and load a sample for us. So, Holly, what sample did you have there, that well plate?
Yeah, it's a well plate, and is Rocio on? Maybe not. Okay, it's actually an organoid sample that's in A1 that has, I think, 4 color fluorescents.
Okay. All right, so we can use this as an initial sample to look at. And we can also maybe change samples to show how other samples would look as well. So let's go ahead and load that sample in. You already saw the placement of the plate on that loading dock with well A1 on the top left. You can then load the sample by either touching the touchscreen. It'll give you that option to load the sample, or you also have that sample here in the software. So if you focus your attention now to the software, you see this load sample icon over here. So let's go ahead and load the sample. Is it ready?
Yeah.
Okay, so I'll just go ahead and load that sample. And then I'm going to minimize this screen and move a couple of things around, just so I know where I'm looking at. And let's see, can I minimize this? I'm trying to, there we go. I'm trying to get rid of this window or make it smaller. That way I can see the controls. Maybe I can hide it. Can I take it somewhere else? Okay, there we go, that's better.
I think that's as small as it gets on Zoom.
Yes, okay, all right, that is perfectly fine. Okay. Let's see. And yes, if anyone has any questions, please feel free to stop me as I go. The idea is, for this to be as helpful as possible for you. So if you have a specific application that you have a question about, feel free to let me know. All right, now I'm going to talk about what the system is doing as we're loading the sample. So I mentioned that the system can do some automatic detection for what your sample is, right? So that's the process that is happening right now. The system is first detecting what your carrier is. And by carrier, I mean if it's a 24-well plate, a 12-well plate, a 6-well plate, a 35-millimeter dish, a slide, it'll detect exactly what that carrier is. And it does this based out of this overview picture that it captures using a camera that basically just takes a snap of the top of your sample. And then it uses AI to actually use that and determine based out of that what your sample might be. So it automatically detected that this is a 24-well plate. And it also did some measurements out of that plate. These measurements include both determining the bottom material and measuring the bottom thickness. So by that, I mean, it detected automatically that this is a glass bottom or optically clear composite plate. So it also would detect if it's, that new generation of thin plastic plates that have the same properties for glass. So it detects the material, it knows the refractive index of that material, and then it also measures the thickness of that material. It also measures the skirt height. The skirt height is essentially that lip that hangs at the bottom of the plate, how plates stack on top of each other thanks to that lip, that skirt. And this is important to measure because the system will use this information to set the travel ranges for your plate in a safe manner so that there's no risk of collision with the sides of your plate. And then obviously the measurement and determination of the material and the thickness that it's going to be imaging through is going to be very useful because the CD7 actually has automatic correction objectives, which will take this information and adapt their optical properties so that they're ready to image through these samples of different thickness of different materials without compromising on image quality. And so that there's no induced spherical aberrations, which is typically what would render an image blurry or hazy if you're trying to image through a material that your objective is not corrected to image through. Most standard objectives, most standard modern objectives are corrected to image through a number 1.5 cover slip. That's why we use that certain number of cover slip for cover slipping, or the 35-millimeter like glass bottom dishes typically have that thickness built because that's what most objectives are corrected to build. However, as you see here, this plate has a slightly different thickness than that, and obviously you might have a lot of different plates, maybe some plastic plates that will definitely have a much different material, a much different thickness. And one of the benefits here of the CD7 is it can actually do these measurements for you and correct the colors automatically for you so that you're ready to image without any further setup time. All of these measurements are done automatically by default. And that's what these check marks here mean in our sample tab. It tells us the information of the sample and gives us the option, if we want to forego these measurements. So for example, if we've already loaded this plate, maybe we're capturing some images, maybe we have a live tissue culture here, and we want to set up an experiment where we add certain reagents and we just go back and forth and image live. If we already determined all of these properties once, maybe we don't need to keep doing it again every single time that we load a different plate in. So in those cases, maybe you might want to disable the determination of the bottom material, the bottom thickness, the sample carrier detection, even the carrier overview if you don't need it. That way, the system basically just will load the sample back and forth without doing any of these measurements again, because it does take maybe a couple of minutes to do these measurements the first time around. Okay, any questions about the automatic sample carrier detection and the measurement that the system does? Okay, you will notice that when you do these steps, if you do all of this sample carrier detection and overview, measure bottom material, measure bottom thickness, the system actually is acquiring a Z-stack of the bottom of your plate when it does this, and that's how it determines that thickness. You might even see this in the display screen, in the LCD screen. When this process is running, it'll actually tell you that it's acquiring a Z-stack and it's doing step, I don't know, 100 out of 250 or something like that, that it takes to capture that Z-stack. So you can always actually see how far into that process live if you look at the LCD screen. Okay. The other thing that I'll mention here in terms of the automatic carrier detection and the carrier overview that is created is you have this, hold on, I'm getting a zoom error here. Is that on my side or is that on your side, Holly?
Oh. Sorry, that was making me nervous.
Okay.
No.
So it's signed outside of Zoom.
Let me try.
And you just reduce it and then it'll just go off the screen.
It's still sharing, right?
Yeah.
It's still sharing?
It looks like it's still sharing. And it looks like everyone is still viewing the screen and we're still recording. So
okay.
I think we're okay.
Okay. Sorry.
All right, no problem.
Awesome.
All right, so what I was about to mention was we do have this carrier overview that gets created for us. This is super useful, not just for well plates, but for all kinds of samples as well. You'll see that we have this sort of quick transmitted light or bright field image of our plate. If you do write annotations on your plate with maybe your different conditions, you can actually use this overview to remind yourself what those conditions are. And this will also be saved in the metadata of your files. So if you have here your conditions set up by columns or by wells and with some writing on it, you'll be able to see this also on your files. It works best with metallic markers. So I left behind one of those metallic markers for you. That way you can use that if you wish to label your plate. And that way you can also remind yourself what your conditions are, where they are, and what they contain. You'll also notice this within the overview, we have this ROI here selected. This is actually where our nose piece is currently located. So you could also use this to help you navigate your sample. So you can say like, okay, I want to go to well A1, and you could just double click on that well, and the system will go to that position, and you can go to any other position that you want here as well. If you have slides, there's a good chance that you'll be able to see here your slide and your cover slip, and maybe even in some cases, you might see your tissue section as well, if you're using some tissue sections. So this could also be very useful to help you navigate and quickly find your sample in this way with this carrier overview. Okay, so I think that covers what I wanted to mention in terms of the automatic carrier detection features, some of the measurements that are done by the system when a new sample is loaded, what the carrier overview is and what it can help us do. So now I'm going to go over a few of the differences here in what we have, some of the control options that we have here on the right side, and some of the control options that we're going to have here on the left side. Let me.
Question in the chat.
Okay, cool.
A1 is staying with Dappy, Alexa 48, 568, and 647. Awesome. Thanks.
Okay, great. All right, just minimize this for now. Okay. All right, so what I was going to do next is now talk a little bit about what we see here within our Zen CellDiscoverer software view. So if you look here on the left side, we have some tabs that might look familiar to you if you're familiar with our Zen systems, with our confocals. You'll see here our acquisition tab, you'll see processing analysis. Many of these will look very similar to what you would find in our confocals. This is where you're going to build your experimental workflows. So for example, in the acquisition tab, this is where we would add or create channels. If we want to do Z-stacking or tiles or a time lapse, we would enable these options. And I'll go through all of this in detail, but essentially this is where you're going to build your acquisition workflow. We also have here a tab for processing. So once you've actually acquired an image and if you want to maybe do deconvolution on it, or maybe you want to create a subset or export an ROI or export a movie, you're going to do all of those options here under your processing tab. You also have an analysis tab. So if you want to create an image analysis routine, which can be maybe some segmentation or some expression, maybe you want to do a cell count or a confluency measurement or an expression measurement, you would go here under image analysis tab to build that routine. And even better, in your own CD7, you actually have a second workstation with direct processing that will enable you to push data to that second workstation. That way you can actually do some processing on the second workstation without burdening your acquisition workstation. That way you can keep acquiring and keep on getting data. And in parallel, you can also do some of your processing at the same time if you wish.
So I booted that the other workstation up, so it's right next to the computer here, and it automatically goes into listing mode, right, when you start Zen.
Awesome. Yeah. So it's designed to be able to communicate directly with the data that is acquired here, and you can push the data to that workstation so that you can do some processing on it as you wish. And you'll also see the same options here. You won't see the sample or acquisition options here, but you will see the processing and analysis as well. Okay. So that's sort of what, in a brief overview, you have here on the left side. I'm also going to talk a little bit about what you have here on the right side. And maybe I'm going to increase my workplace zoom a little bit. That way everyone can see this a little bit better from their laptops. This essentially, we call it the aging slider. So as we get older, we might need to slide this further along so we can actually see this and read this a little bit easier. Feel free to revert this back to a comfortable setting on your own workstation. But for everyone joining from our laptop, this might be an easier view. So on the right side, what we're going to have here is first a container that is going to show you all of your current open images. So right now we don't really have anything that we acquired. We see the overview, the carrier overview that was created automatically as an image itself, because it is an image and we can save it. And again, it will be saved in the metadata of the files too anyway. However, what I wanted to point out here is anything here on under or below the CellDiscoverer window, this is actually your microscope controls. So this is where you get a chance to choose your objectives. You can control the movement of your stage. So if I move this, you'll notice the crosshair here moving as well. So this is letting you control the stage in X and Y. You can also control your stage in Z, so you can move up and down in Z if you wish. You can also use our focusing routines or incubation. And I'll go into detail about what each of these are. But for now, what I want you to be aware of is that on the right-hand side, You're going to have all of your active microscope controls. So think about this as your digital touchscreen that you had on the confocals, right? Where you could choose in there the objective, maybe the channel, maybe turn on transmitter light, turn on reflector light, whatever that might be. You now have those controls here in the same software. In contrast, if you're actually building your acquisition workflow, that's all going to be on the left-hand side. So again, this is where you're going to come in to build your channels. If you want to build in a focus strategy for your workflow, you would do that here. And this is actually how the acquisition will take place. And I'll show you what that means, how that works. Okay. All right, so now I'm going to spend a little bit more time about describing the control options for the CD7. As you can see here, the CD7 has these 4 objectives, and actually it has more than that, than just these 4 options. The CD7 has a unique optical design where it has a magnification changer that can multiply this nominal magnification for each objective by either 0.5, 1x, or 2x. This means that, for example, my 5x objective can be either a 2.5x, a 5x, or a 10x. Now, this is not just adding a piece of glass into the light path. This is actually changing the light path and changing the tube lengths altogether. So there's actually 3 different tube lenses in the microscope, each with this adaptation. This means that you do get a different optical property at different level of resolution with each combination. So for example, the 2.5x option has a particular NA, I think is of 0.15, I want to say. The 5x combination has a higher NA, I think it's 0.2 or 0.25. And the 10x combination has the highest numerical aperture combination here of 0.35, which is what's underneath the objective. And numerical aperture, for some of you that maybe haven't heard that term before, it's essentially a measurement of the level of resolution that that objective can achieve. And typically, the higher the NA, the better the lens in terms of the level of resolution and light that it can capture. So a higher combination with a higher NA will give you a higher resolution image. In contrast, a lower combination will give you a lower resolution image, but it also will have a much wider field of view. And we can see this here if I go to my navigation tab. You'll see that my 2.5x has a pretty wide field of view, but if I go to my 10x, that will obviously translate to a much smaller field of view. So even though I'm going at higher resolution, I'm probably going to need some tiling or something if I want to capture an area as big as I had here with my 2.5x. Now, all of our objectives are very high resolution objectives as it is. So even the 2.5x is still a great level of resolution that can, for example, help you screen a 96 watt plate or a 384 watt plate, and maybe just take one picture per well to capture most of the well, but still be able to get data out of that in terms of maybe doing a nuclear segmentation or an expression measurement. So this essentially means that these 4 objectives can actually turn into 12 different magnification combinations. And I'll spend a little bit of time just talking about what the different objectives are and what they're best suited for. Our 5x objective is a lower magnification objective. It also has the longest working distance, and it's really incredibly long. This 5x objective can go all the way up to I think it's 5.2 millimeters in working distance. This means that this objective can go through thick plastic plates, which are typically 1.2 millimeters thick. Or, for example, if you have hydrogels and cells embedded in hydrogels, this objective will also be able to get deeper into those hydrogels because of that 5 millimeter working distance. The working distance stays constant at all combinations. So you can have 2.5x, 5x, and even 10x with that 5 millimeter working distance. So for example, for today, for the organoid, depending on its size, this could be a very useful objective to image the entirety of that organoid if the organoid is a thick organoid. Or also if we were imaging through plastic plates, this objective would also be able to get us a good image through those thicker plastic plates. Our second objective here, our 20x.7, is also a long working distance objective with a working distance of 2.2 millimeters. But it's also an incredibly high working distance for a 20x. And again, this means that we can get, with this combination here, we can get either a 10x, a 20x, or a 40x objective, all with that long 2.2 millimeter working distance. So this objective is also going to be suitable for imaging through plastic or through hydrogels or larger 3D samples like larger organoids, maybe small animal models, that would require that longer working distance to be able to image the entire thing. And obviously, this comes with an increase in resolution with up to 0.7 numerical aperture when used in combination with the 2x magnification changer. So this would be at 40x a 0.7 NA objective. At 10x, I believe the NA is actually the same as our 5x.35. However, the 20x does have a little bit better of light gathering capabilities. So this 10x combination typically will give you a better signal compared to the 5x 2 times combination. You'll also notice that when I call this objective into play, it gave me the correction that is being applied to your sample. So again, this information was acquired during the sample detection. It detected that it was a glass bottom plate with this thickness, and it adjusted the correction color to match and to correct that color specifically for your sample, and it did all of this automatically for you. This objective, because of its long working distance, can also correct for both thin and thick plastic plates. This is PS for polystyrene. Okay. Our second 20x objective, 20x.95, has a higher numerical aperture of 0.95. This means that you could image at 40x with 0.95 NA. That's an incredibly high NA. And this one also has a low working distance, but not quite up to 2.2. I think the working distance of this objective is capped at, I want to say, is 0.75 millimeters. So it's still pretty long, but not enough to get through plastic plates, right? Because again, those are typically 1.2 millimeters or so. That means that this objective is going to be better suited to image through glass. So that's why you see here that this objective can correct itself to work with either thin glass or thin plastic plates, and it can correct the spherical aberration up to a thickness of 210 microns. Again, this will allow us to use this numerical aperture and working distance at 10x, 20x, and 40x. Now, last, we have our highest numerical aperture objective, our 50x. The 50x is an automatic water immersion objective. So whenever you click on it and it's called into play, the system will give us this little window here where it tells us immersion is currently being applied. That means that the system is applying that little bit of water needed to create the immersion in between the objective and the plate so that it is ready to image. And it does this all automatically. This 50x has a working distance of about 0.85, I want to say, if I'm not mistaken. It's just a little bit higher, actually, than the working distance of our 20x, and also suitable for imaging through glass or through optically clear composite plastics or through thin polystyrene plastics. The particular thing about the 1.2 that's a little bit different to the other 3 objectives is that because it's a water immersion objective, its numerical aperture of 1.2 is fixed at all combinations. That means that you can have 25x 1.2, 50x 1.2, 100x 1.2, which hints that this combination here, 25x, it's a super strong combination objective because it essentially gives you the fields of view of a 25x, but with the resolution capabilities of a 50x, 1.2 numerical aperture. So that's a really fantastic combination that will get you really incredible high level of detail for your samples. And again, this objective works only with glass or thin plastic plates. The system is smart, so it'll know that if you loaded, for example, a 6-well tissue culture plate that's 1.2 millimeters, if you try to use one of these two objectives, it will give you a warning and it'll let you know that these objectives are not suitable for imaging through that thick material. All right. Any questions about the objective options in the CD7 and what they're better suited for? Okay. Next, you also have here your stage control, which again, like I mentioned, can help you move and navigate around your sample. I'll show you how we can use this to help us navigate here. This essentially acts as a virtual joystick that can help you move your sample in X and Y. If you want to move very finely, you can just go up to the inner circle, or if you want to go faster, you can go all the way to the right. Or if you right click on the joystick, it'll turn red and then it'll move much faster. You could also physically input an X and Y position if you want to. So if you mark the position that you want and you want to return to that, you could also write that in as well. You can even control the move speed of your stage by using here your speed and acceleration options. Typically for air objectives, maybe I'll go back to our low magnification objective here. And you'll see that whenever I do that, the system will also automatically remove my water immersion for me. And I'll have to wait just a few seconds for that water immersion to be suctioned off before I can proceed. However, the good thing about this is because it's water and not oil, I can go back and forth between different magnification objectives and not have to worry about having to take my sample out and clean it every single time. Also, because the water immersion works for 25, 50, and 100x, I can potentially, start an acquisition at 25x with water immersion. And if I want to zoom in further, I can go to my 50x and I won't lose my spot where I'm imaging because the water immersion is not getting disturbed. It's going to still stay in place. That way I can switch between 25, 50, or 100x and always be within my same field of view. Okay. You'll notice here again the speed and acceleration options for your stage movement. So if you want to move your stage, it'll move at this speed. We could also bake this into an experimental profile. So if you have some samples and you're worried that they might move around or jostle around too much and you want to be more gentle, you can always decrease the speed and the acceleration of the stage. This again will let you control the Z position for your imaging. I'll show you another option of how to do that as well once we go live. And then this is our definite focus, which is a hardware-based function that will find the surface of your plate. And by the surface of your plate, I mean the top of the bottom of your plate. So it would essentially find that interface in between the aqueous media and the glass plate. So again, the top surface of the bottom of the plate. This is typically for most samples that are, monolayers on the plate. This is exactly where your sample is going to be. So this is going to be a very useful tool because it's not even a software-based autofocus technique. It's actually a hardware-based way of finding that position, and it works really well, and it's very fast. So it can help us find our samples very quickly. And then I'll also mention here that you have your control of your incubation. So if you do want to turn on incubation control for your experiment, then you'll want to turn on heating here, and you can set the same point. Obviously, for most cell culture, you'll just keep this at 37°C. And you'll also want to turn on the CO2, which is by default set here to 5%. And again, before you do this, you do want to make sure that you turn on the or open the the tank. It should already be sort of preset at a specific pressure in the manifold to send that gas line into the CD7. And when you turn it on, you do also have to turn the air circulation within the chamber on, and a setting of 2 is great. For most applications, this is where you're going to be, just level of 2 for the air circulation within the chamber.
Sebastian, do you know what the tank regulator range should be? Should be like,
I believe it should be between 10 to 15 psi.
sorry?
I believe it should be 10 to 15 psi.
Okay, thanks. I'll just note that on the regulator.
Yeah, and typically what I'd recommend is you can just set the manifold to that outgoing pressure. That way, whenever you open the tank, all you have to do is just open the tank and it should be already preset to that pressure.
That's exactly how we do it.
Perfect. Okay, so that would be your incubation control. The chamber inside of the CD7 is actually pretty small, so the temperature will stabilize fairly quickly. However, if you are doing a time-lapse experiment, you want to be imaging, for example, the same position, something like, let's say, every 5 minutes for 2 hours. I do recommend that you maybe place your sample in and turn the heating on and let it equilibrate for 15 minutes to half an hour, because the chamber will actually equilibrate pretty quickly, but your sample might actually take a little bit longer to equilibrate. And while it equilibrates, you might see some fluctuation in the sample. So obviously, if you want to be capturing that same spot continuously during your time lapse, you might want to make sure that your sample is also equilibrated. Okay, and then lastly, we have here our auto water immersion control. As you saw, whenever I call the 50x objective, and maybe we'll do it here again, the water immersion is applied automatically. However, maybe it might give you a message where it says that it couldn't create water immersion. It could be that maybe the plate is not completely flat, maybe it's a little bit tilted, so you might want to check that. Or it could be that maybe there was an air bubble in the immersion line. And in that case, you would basically just reset the immersion, and you can do it manually by using the control here on the bottom. I'll show you how that works. You can essentially just tell the system to remove the immersion and apply it again if needed. And that's what you would do with this control here, where you can have the option of removing that immersion. Okay, so I think that covers all of the actual microscope controls.
Actually, Sebastian, it's not very clear on Mrs. Feather. When you said there could be a bubble and we'd have to do it manually, can you actually be more detailed with that?
Sure. So if you're calling in the 50x and you ever see an error that says something like water immersion could not be applied.
Yes, I got all that.
There could be a couple of reasons for that. Actually, the most common one that I've seen is that the sample is in the implant. So maybe the sample got loaded slightly skewed, maybe the plate is slightly tilted. I've seen it also on slides where the slide was kind of loose on the holder, and then couldn't create water immersion. I've also seen it where maybe the immersion hasn't been used in a while. So it could be possible that some of the water in the line evaporated, or a bubble got sucked inside of the immersion tube. Yes. And then that could also cause the immersion to fail because it tried to apply water and there wasn't any water, there were just bubbles. So to sort of purge the line, if there are bubbles in the line, you would click here on remove immersion and then click back on create immersion. And then you could repeat that process a couple of times, and the system will let you know if you was successful at applying the immersion or not. Okay. If you keep having the issue, then actually the chances might be with the sample that maybe is tilted. So then at that point, I would just eject the sample, take a look at it, make sure that everything was right, and then place it back in.
Okay, but this should fix it. And if there's ever a point where it won't fix and it's not the sample, is that something that we on our end would fix, or is that like a service issue?
You can contact us at service. I haven't.
We haven't come across that.
In most of the cases that I've seen, it's been a problem either with bubbles in the line or with a sample issue.
Okay.
Yeah, one of the for example, one of the last things that I saw where there was an issue was the plate that was definitely tilted, and if it's tilted, obviously the immersion would not apply properly. Or I've also seen it where a user was using glass slides with the cover slip on it, but the cover slip wasn't sealed. So actually, when the immersion was applied, it sort of suctioned off the seal, the cover slip. So obviously, it can't apply immersion properly because the slide was sort of just moving around.
So I have a question. This is Mary.
Okay. Yes.
Yeah, I was wondering if there's a specific type of water you recommend to make sure it doesn't have high ion concentration or something like that to protect the lens.
Yes. So the immersion fluid we recommend to be DI water, and that's also the same recommendation that we have for the humidifier tank. It says right on the bottle itself as well.
Okay, thanks.
Okay. All right. So that was it in terms of the water immersion. I'm going to revert back here to a low magnification objective. And then I also want to point out here my navigation tab. I didn't quite talk about this when the sample was loaded. But this navigation view was created automatically for me, matching the automatic detection of the well plate format. So again, the system detected that it was a 24 well plate. It set up my 24 well plate layout. You'll also notice that it set up these boundaries for me. And these are color coded to match our objectives. They are actually stage travel limits that are set to protect your sample and the objectives from collision. And this is based on the lip or the skirt that was measured for each of your samples. So this is actually a fairly low profile skirt height, which allows us to use all objectives, pretty much almost the entirety of the plate, with maybe just a few edges missing. And again, this is just preventing collision with that lip or that edge on the side. So even if I was going to try to navigate there, the system will stop and it won't let me navigate outside of that safety travel range. That way there's no risk of collision at all with your samples. Like I mentioned, this is color coded. So you can see here the travel limit for the red or 5x objective, which is our longest working distance, the most shallow profile. So obviously it has the longest working travel range as well. And then you can see here the limit for our 20x in green and for our 50x in blue. Now the 20x in yellow overlaps with one of these. So if you don't see it, you could just select it and it will just show you that color. It was basically overlaid with our 5x objective, again, because it's also a long working distance, 20x. So that means that even at 40x, I can image pretty much the entirety of this plate. You might want to be more careful for, say, 96 well plates, where depending on the skirt height, some skirt heights are considerably long. I've seen some up to 3 millimeters thick. And then that is going to limit our travel range to those outer columns and rows, more so for these smaller well formats. And this is something that all systems are going to have, that limitation of maybe not being able to image all the way to the outer wells. The difference is the CD7 will be smart about it and detect those limits, so it doesn't accidentally let you travel out to those ranges, while in other microscopes, you might just hit the plate without realizing it. Okay, so now let's look at our navigation view setup, which is made to help us create our workflow within our sample. And let's look at how do we create those workflows. I'm going to start with using our Widefield modality, so camera-based acquisition. For most of the description of how to do Z-stack styles, time series, how to do some automation, but all of that translates exactly the same if you were using a confocal track instead. I'll just use Widefield for now to show you a little bit faster how those options work. And then at the end, I'll also show you how to set up and optimize confocal and Airyscan tracks as well. All right, so first thing I'll show is how to create channels. The easiest way to do so is by using here our smart setup. And this will create a new workflow with either this experiment name here, or if you've already had a name, you could also select a workflow that was previously created, or you might just select a new workflow. So for today, maybe I'll just call this is 24 well organoid imaging. And the workflow will also remember the format of the plate. So that's why I'm putting here the 24 well. And maybe let's start with this being a Widefield workflow. So Widefield 24 well organoid. And then we can use the smart setup to help us create our channels. So when you click on the smart setup, it'll first ask you if you want to build confocal tracks or Widefield tracks. And actually, you could even build both all together. I'll show you how that works as well. But for now, we're just going to build some Widefield tracks initially. I'm not going to add just a few channels here. I believe I read in the chat that this sample had DAPI 488. I want to say it was 568.
568 and 647.
And 647. So you can just use this search feature here to look for the dyes in the database. We also have a list of recently used dyes. And then we also have some transmitted light contrasting methods that are great in the CD7, and I'll show you what those options are as well. I'm going to add our oblique imaging technique, which is my favorite in the CD7. And I'll also add here phase gradient as well. So here I have a combination of fluorescence channels and some transmitted light channels. I can also select the pseudo coloring for my channels here. So maybe let's make the 647 magenta so that it's different from that 568 in red. You'll see that the smart setup sort of recommends a setting for me. For Widefield, it'll just give me one option. But when you're in confocal mode, you'll actually have some different options here that will prioritize either the signal or speed of acquisition. And I'll show you what those options are when we go into confocal imaging. For now, let's proceed with our Widefield acquisition. So I just used that smart setup to create these tracks for me, these Widefield channels. Again, this is camera-based acquisition, and it's using our Colibri 7 LED. So each channel will have its own excitation source, and this is all using LED-based excitation. And it calls the right LED color that I need for each of these channels. You see here the selection that was created automatically. For transmitted light, I obviously don't have that Colibri 7 LED control, but I have control here of my transmitted lamp, which actually is another LED, and it's not even a white LED. It's actually a near IR LED 735 range LED designed to be able to do transmitted light imaging in a gentle way for live cell imaging. So without inducing the phototoxicity that we've seen caused by blue wavelengths, right? Typically, blue and UV wavelengths induce phototoxicity more than far red wavelengths. So we designed our transmitted light to actually work with a near IR gentle wavelength to do our transmitted light imaging.
Wait, sorry, so both of those use the near IR?
Yes, all transmitted light imaging in the CD7 uses transmitted near IR. This is also why in Widefield, we don't have an option of adding filters for example, like Alexa 750. And that's because that wavelength is occupied by our transmitted light image technique. Okay. So let's take a look at our sample. So I've added here some channels. I'm going to navigate to well A1 since that's where our sample is located. I'm just double clicking here on well A1 and I can go live, and there's a few options or where I can see my live screen. I can either have it go into a new tab altogether, view it as a separate container, or view it within my navigation view. I'll leave it as a separate container here for now. And let's go live here and see what we see. It looks like this might be our organoids here. I'm clearly overexposed, so maybe I'm just gonna reduce here my LED power output and maybe optimize my exposure time here by just clicking on set exposure. This will automatically find an optimal exposure setting for me to fill 35% of my dynamic range in my camera. That's what this 30% here means. And this just makes it so that I have 30% of my dynamic range filled. That way I can have plenty of signal to look at my samples, but also have enough dynamic range left. So if there's a brighter spot somewhere in my sample, I can still capture it. And this is what's recommended by default for all fluorescent applications here in Widefield, to configure a 30% shift. That's what this set exposure will automatically do for me. I haven't actually focused at all. So let me use here my find surface tool. That way you also get a chance to see how that works. You will essentially do a physical way of finding where the bottom of my plate is. And it determined that this is the bottom of my plate. So for most instances, if your samples are at the bottom or surface of your plate, it'll take you right to your sample already. And then we can always critically focus by controlling the stage, like I showed you here, where you can move the position of the Z-plane in or out of focus, or how I prefer to do it, which is by pressing the control on your keyboard, and then you can control the step size. Let's move kind of slowly here, so that every click of your scroll wheel on the mouse, so if you use the scroll wheel on the mouse, now we have control of critically focusing by using our mouse as well. So we can move in and out of focus using the scroll wheel. This is another way of moving our focus in and out. Okay. One thing that I really like doing in the CD7 when I have a new sample, and maybe I'm just getting my bearings around what the sample might look like, is I might just see my live view purely in my navigation view here. So as opposed to the separate container, which opened that container on the side to show me, I can just view here with my my navigation tool, and I can even move my ROI around so I can look at different locations here within my well. Or what I can do is, if I found, a relative location of where my sample is, maybe I'll, critically focus in a little bit just to check out what my distribution of my samples look like. I could maybe even let's just use DAPI only for now. I'm going to deactivate my other tracks. There we go. And if I want to get a quick idea of what my well distribution looks like, I can just quickly here take a quick tile snap. Let's do a 3x3 here for now. Quick and simple. It'll automatically acquire this fast tile centered around where I was currently located. And I can get a quick idea of the distribution of my sample within that well. Here, I'll also point out that I could image even without these limits that are set here in my, my well plate. This is just an approximation based on the knowledge that this is a standard 24 well plate. If we did want to be super accurate and know exactly where our well edge is, we can run a process to calibrate the well plate, because obviously every brand will be a little bit different in terms of the size of the wells, the location of the wells. We can always do that step if we want to, know exactly where the edges of our wells are. But this is super useful here to just get an idea of the distribution of our sample. Also works really well for slides. So you have a tissue on a slide. So the first thing that I typically do is just turn on DAPI and get a quick tel image of that slide to get my bearings around. And now I can use this to navigate. So if this was a particular, sample that I wanted to image, it's like, okay, let's see, this one looks pretty. So I could just double click on that, and then it will center on that particular sample for me to look at. And that was just by taking that quick 3x3 snap, which is saved as an image itself. So I could always actually save this data altogether. Okay. Let's look at this in maybe a little bit more detail. Maybe I'm going to go here and let's call our 20x objective. So I'm going to go here because this is like last bottom plate. I can use either my 20x or my 50x. You'll see that my field of view adapts to the size of my view. So here I have a 10x field of view. If I selected my 20x, it would now be condensed to my 20x field of view. So let's look at here at how we can optimize our imaging parameters for this sample, or maybe we can choose. Yeah, that looks like a good sample. There's obviously plenty of samples around. So what I might want to do now that I'm at this 20x combination is again optimize my exposure settings, right? So I don't even have to go live for this. I can literally just go by channel and click here on set exposure. It will automatically find a good exposure setting for me at each of these for each of these channels. Okay, it seems like the intensity for 568 is a little bit lower. We'll take a look at what that looks like. I could increase the LED power output if necessary, if I want to get that channel. For now, it seems like these three are fairly bright. So let's take a look at what we have here. I'm going to go live again. And maybe I might need to critically focus now here that I am at 20x. And again, I can just press Control on my keyboard and use the scroll wheel on my mouse if I want to critically focus. Okay. That looks like we're getting there. We might already be in the middle of the organoid. Obviously, we can see a few different layers here. Okay, so we are where our organoid is. If we wanted to, we could always at any point just take a snap, and this essentially just takes a picture with whatever channels you have configured in the exact location that you have. So for example, that snap here captured these three channels here automatically. And again, I didn't even go live to look at any of them. I just used my set exposure setting here to find a good exposure setting for each of these three channels, which is that 30% shift. And then I can just press min max display to bring that display setting to a nice setting. I could even pre-build this display adjustment into each channel. That way, every time I take a picture, it will automatically apply that display setting for me. We can take a look into the red expression if we want to. Maybe it might be a weaker expression, or maybe it's a rare expression, or maybe not organoids, all organoids are expressing it. That is completely fine. And actually what I could do is go back to my low magnification overview that I acquired here And I could add all of the channels in here. So that way I could have an overlay of DAPI, green, 647, 568. And then based on that, I could use that to maybe navigate to my actual organoids of interest. So if there was, some of them that were expressing certain proteins more than other, I could quickly take a look here with that overview and then go to those or to a higher magnification to image it in detail. Okay, so let's look at, now that we've seen how to create channels for acquisition, let's look at how do we create or add Z-stacks, because obviously these would need a Z-stack image to be able to image the entire thing. And maybe let me go to a smaller one here for demonstration purposes. If you want to enable multidimensional acquisition, you have the options here. So I'm going to add here a Z-stack, and this will look, almost exactly the same as how it will look in your confocals, right? Where you have the option of setting a first and a last C-plane. It'll give you a range, the number of slices. You can set your interval. You can choose the optimal interval, which is what I always recommend. Or you could also just set a center position and then arrange around that center. Let's use here the traditional first and last method, where I can, right now I had selected my green channel. That's fine. I could do it in DAPI as well. And I can always just scroll in one direction in Z. Maybe I'm going towards the top of the sample here. And let's say, okay, that's probably good enough as my first plane. So I can click here on set first, and then I can go the opposite way and scroll through my sample to find the bottom. And you can see even with Widefield, I can get a pretty good level of detail here. And I haven't even gone into the confocal. Obviously, we're going to be limited here by what the Widefield can optically section, but it'll give us a good idea of the setup for now. So let's say that, and maybe we don't need to capture the whole thing. Maybe let me go a little bit further back in. Let's say that that was the last position that we wanted to capture. So that means that we have this range that ended up being close to 60 microns, which would be at a 1 micron interval, 60 slices. Again, I usually recommend to use the optimal setting. It would obviously increase your number of slices, but it will give you better optical sectioning. And this is true still even for Widefield, obviously even more so for confocal. This is really where it's important to use that, because then you do have the optical sectioning with the confocal. But even in Widefield, this is useful because if you want to use something like deconvolution to clean up your images, then it'll work way better if you actually use the optimal sampling step. If all we wanted to do was set up a Z-stack, then that would be sufficient. And I can always, maybe double check that my channels are optimally set up. So I can always, just go back to each channel and just select it and see how that signal looks like. Maybe I'll check out my red channel again and see. There's a little bit of signal in there, but it's definitely more faint. So maybe let's just leave that red channel out for now. But if we were ready to image now, then we essentially would just click here on Start Experiment, and it will capture that full Z-stack for us. When you do that, it will give you a live view of all of the steps that are imaging. It'll also tell you how long that acquisition is going to take to acquire. So you can see here, it's going to take just a little bit over a minute for those, how many? 123 Z steps. So it's actually really fast. And obviously, that's partially thanks to us using that Widefield camera mode for now, just to show the setup of the experiment. Okay, any questions so far about how to create channels in Widefield, how to set up a Z-stack acquisition? Okay, so next I'll show let's wait for this to finish, just a few seconds. Maybe I'll try to do a deconvolution here real quick on this as well, just to show you some processing options. But next, I'm going to show how to create tiles and positions and how to add time series for time-lapse experiments. Okay, so we have our acquisition finished. The signal is fairly bright, so I might actually revert back my display adjustment a little bit so it doesn't look so bright for us here. Maybe something like this might be a little bit nicer. And by the way, I guess I'm doing here, just so you know, these display adjustments are like just like that. They're just display adjustments. They're not actually changing your data. They're just changing how we display the data. This is a very useful feature here. I could even change this color. Like, for example, if I wanted to make this red again, I could just do that here as well. Okay, I can always scroll through my acquisition in Z. So maybe I'm gonna zoom in a little bit and show you. So I can go from the top Z, and then I can scroll through the whole thing in steps. So I have that availability here. I could also visualize things in 3D. This being a wide field image might not have the best optical sectioning here, but it's still going to be a good visualization. And obviously this will work really beautifully with confocal data. So you'll have the option to here visualize your data in 3D as well. And you can always rotate the data here. We ended up acquiring only about half of our organoid in this particular case. The orthogonal view is also very useful, very well used, because it sort of gives you that cross-section overview from both sides and also the current Z-plane that you're in. And you can always scroll either in Z or in X and Y to look at different areas of your cross-sections. For wide field images, like you saw, it was super fast to acquire that Z-stack, right? It took like less than a minute and a half. So it's a very useful technique if speed is of the essence. And you could still do some optical sectioning in this case using a software approach like deconvolution to clean up the image a little bit. And you do have a few different options here of how to use that. The best one is definitely constrained iterative, but it is also the slowest, so it does take a little more time. You do have a few other options that are a little bit faster. Maybe for demonstration purposes, I can do maybe not the best one, but one that's maybe a little bit faster than that. You would just choose your control. You actually do have the option of even choosing different strength methods for different channels depending on the method that you're using. But for the most part, you can just use some of these presets as you test this, and then you just click on apply and you apply the deconvolution process to clean up that image. While that takes place, maybe I'll also go back here and now mention some other options that you have available in wide field imaging, which I haven't shown yet, and that would be our oblique and PGC technique. These techniques are transmitted light techniques. Like I mentioned, they use a near IR LED. Okay, let's see, that was our deconvolution processing. Okay, might need to try deconvolving that again because it didn't work for all channels. We can take a look at that a little bit later. Just a processing step. And the data set was a little bit bigger than what I would typically like to deconvolve. It should still work, but maybe we can revisit that at the end if we have time. Okay, we have here now our transmitted light technique. And here I'm just looking at oblique. Like you can see that this oblique technique really gives really great level of detail, especially for samples that have some three-dimensional aspect to it, because it illuminates the sample at an angle, which creates this really great contrast. It can also be used to acquire in Z-stack. And if you do it, you'll actually be able to scroll through the Z-stack like I'm doing here and be able to visualize different details. And because it's a label-free technique, it'll essentially just give you an idea of the whole contour of your sample, even if in areas that maybe don't have fluorescence. So we have here our oblique technique. We can do the same thing as before, where you would click here on set exposure to find an optimal exposure setting. For oblique, we actually recommend a 70% shift as opposed to the 30% for fluorescence. And maybe I'll capture a quick snap here for both oblique and PGC and talk a little bit about the differences in between the two of them. Okay, let me open this here, just show one channel at a time. So we have here our oblique technique, which is essentially one single angle of oblique illumination. And you can see the contrast here that we get from this 3D sample here. Or we have PGC, which is essentially two oblique pictures superimposed together, which can give us different level of detail depending on the angle that we're acquiring it from. I actually tend to like oblique the best because it's a faster image. It only takes one picture and usually does give you pretty good level of detail. And even in 2D samples like cells, it actually works super well and will show you the whole outline of the cell. So if you want to do some live cell imaging and maybe you have like a nuclear stain or something else, but you don't have anything to see the outline of the cell, you can just use that oblique technique and it'll show you a really nice contrast from the get-go. The other cool thing about these two techniques is they are not affected by edge effects. So if you have a 96 well plate and you want to maybe image the entire well plate, you can do that and get all the way to the edges of each well without having that halo or edge effect that you see if you were using traditional DIC or phase imaging. Okay. Any questions about these two techniques or Widefield setups or Z-stack? Okay. I'll show you now. I'm going to go back here to our navigation view. And you can see here, because I acquire another image, it sort of gets added here to my display setting. I can always remove it if I don't want it. In this case, because it has the superimposition of the two of them, it looks basically completely white. Let's go now into how to add tiles and positions into my acquisition. So let's say that we weren't imaging just particular events one at a time, but we wanted to automatically image multiple wells altogether. So you can use the carrier overview that's created for you by the CD7 to create your tiles and positions. Yes.
Hi, Mary, I just want to this is when he's going to move to other wells in case you don't want him going into the other wells for imaging.
Rocio, are you on? That was what she was asking about. I wonder if with the other wells, you might choose areas where there aren't any cells or something like that, because it is good to learn that.
Well, yeah, no, yeah, you have to. But I want to
I
put
don't-
the sample. Is that Rocio's deck? She wanted that?
She had imaged, she wants to image later. And
Okay.
I've feel like she doesn't want them exposed, but I wonder if we could like, I don't know.
So,
stick to a-
I mean, I don't even have to like actually image the other wells. I can just show the setup.
let's do that. I mean, she shows that she's attending, but haven't heard from her, so.
Okay. Yeah, we won't image. We won't image other wells. I can just show you how the setup works.
Okay, she
Oh,
said
so
it's okay to
Rusty-
image them.
Yeah, yeah.
Okay.
Okay, thanks.
So, all right. So let's imagine that we were adding tiles. I'm going to deactivate Z-stack for now, even though I'll show you in a little bit later how we can obviously mix multi-model acquisition in many different ways. So we could add a Z-stack on top of tiling, on top of time series. So that would be like if we were imaging these organoids live and there were multiple organoids throughout the entire plate, and obviously we had to capture a Z-stack for them, we could do all of those 3 dimensions in one single experimental workflow. But let's work with one element at a time. So I'm just going to activate tiles for now. So you see, when I activated tiles, I have this menu come up here on the top and the left side of my navigation view. This is going to help me create those tiles and positions. Also, under my multimodal, multi-dimensional acquisition view, I now have this tiles window that came up again when I activate that tiles option here. Now, what is the difference between tiles and positions? Positions is the easiest one to think about. That is essentially, let's say that we have monolayers of cells in our wells, and our experimental workflow required us to image 3 fields of view per well for the entire plate. So in that case, we would want to create a position and we create, let's say, those 3 positions per well, which we can either distribute randomly, and it would just create those 3 positions per well, or we could have them sort of more organized, set up so that we could have them maybe, let's bias them towards the center, maybe we can do 4. And then that way they're sort of set up nicely distributed within the plate. So positions, we're capturing single fields of view that we can do either 1, 2, 3, 4, whatever number of fields of view you wish to acquire within each well. I did the setup here using the carrier, underlying sample carrier. That way the system is using the information, the knowledge that this is a 24 well plate, and based on that, it is distributing my positions on that 24 well plate. That's what this underlying carrier means. You can also add positions freehand. So you could just say, okay, I'm just going to add some positions where I know I have samples. So we've acquired our overview here, and you can just select the plus icon here. They can be like, okay, add a position here, add a position here, add a position here, and add one here. So in this case, I'm basically just manually selecting where to create my positions based on an overview, or wherever I might see that my sample is located. Now, because these are positions, I'm only remembering essentially the X, Y, and Z coordinate of the center. And you can always see them here. We have position 1, position 2. If we were more someday, you would see also the selection of that position 1, 2, 3, 4 with their particular X, Y, and Z. If we change magnification, so for example, let me go here from this 20x field of view to my 10x field of view. That position is essentially going to double in size because again, I'm remembering only the position and not the area around it, not the tile. So the system will adapt and it will maintain that position there, but my field of view acquired is going to be larger, right? According to whichever magnification I choose. So that's what positions mean. And again, I can create them manually by just using here my location with the mouse cursor. Let me delete all of these Again, I could also create arrays. So I could draw a shape and I could tell it within that shape, create a certain number of positions. So here I have 4 positions within this shape. So I just drew this rectangular area and I just created those 4 positions inside of there. That would be a position array here. So we have here the array shown. It's array number 1 contains 4 positions and each of those has specific position location. And then the last option here, like I showed you before, was how to create positions based on the underlying carrier. Yeah, you can delete the previous positions that I made. And again, this is where you can select the underlying sample carrier, where you can select either on my navigation view or in this navigation little window here. And then you can select the wells that you want to create those positions in and how many positions you want to create them with a bias or with no bias and randomly or evenly distributed. Okay. In contrast, and again, these are created as position arrays, right? Because they're an array within each well. So that's why you see here 3 position arrays, each of these containing 4 specific positions. I'm just going to select them again and delete them for now. The difference between a tiled region is in a tiled region, we're actually delimiting not the X and Y of the center of that position that we're going to acquire. Here, we're going to delimit an area. So for example, if we wanted to literally just draw an area that we wanted to tile. So let's imagine that this was an actual piece of tissue, right? And we wanted to image the entire piece of tissue here. We could draw a specific shape. Let me go in here. Okay, draw a contour. And then based on that contour, it'll create a tile region now, whereas you can see I'm not creating an individual position, but I'm tiling that region to encompass that contour that I drew. So I drew here a circle that I can change the circle shape and size, and you can see that the number of tiles adapt to that area. Also, if I change magnification here, you can see that at this magnification, I have 36 tiles. If I were to go at 40x, I'm essentially going to quadruple that number. So now I have 134 tiles that will be required at 40x to acquire that same field of view. So that's the difference between tile regions and positions, where the tile region will be for an area that you delimit. And you have a similar set of options here where you can either draw the region that you want to capture. So in this case, I draw this circle. You could also just draw a rectangle instead, if that's your choice. Or let me delete this and then also show you the other options that we have here. The contour options, yeah, like I mentioned, you can draw different shapes, so rectangular, or you can do a freehand shape tool. This is very useful for slides and tissue sections as well. So you could do your overview here of the entire slide if you wish. And then based off an overview, you could be like, okay, this is exactly the section of my tissue that I want to image. So you could just draw around. And then when you right click, that shape closes. And then that's the region that you're going to image. And if you made a mistake, you can always feel free to edit it, maybe make it a little bit bigger or edit the points to move things around. Now you also have the option, this is the other way that I use this a lot, and that is to create tile regions based on the underlying sample carrier. So similar to before, we can select our entire plate or maybe just a few wells that we want to image. And we can say, okay, I want to image just 25% of each well, and that's enough. So in that case, you just click here on the plus button and it'll create a region that will be equivalent to 25% of my Crl plate. Again, if you change your magnification here, the number of tiles will adjust based on that field of view. Obviously, the lower the magnification, the less tiles that you acquire. This would be the easiest way if you wanted to image a certain portion of your tissue, of your area, of your weld plate, or if you wanted to image your entire weld plate. I typically find that you don't need to go to 100%, usually about a 75% fill ends up filling up the majority of your weld plate. Okay, so that would be how to create tiles and positions. Now, let me create just 2 dummy positions here, just as a demonstration tool. I'm just going to add a position. Let's say this one looks pretty, and let's say maybe this one also looks pretty. So I'll create these 2 positions at the center of those 2 organoids. This goes for both tiles and positions. The tiles and positions get created, like I mentioned, at a specific X and Y. So that's our X and Y on the plate, and our current Z value. So this is our current Z where we are located. If you're going to do this tiling or positioning automatically for the entire weld plate, you might not want to verify all of those Z positions yourself, right? Because that would take a long time. The idea is that the CD7 can use the automation to verify or focus on those positions for you. And that is what is going to be done here by our focus strategy. We're going to spend quite a bit of time in here because there are a few different options that are suitable for different applications. And to me, this is the most important part of automation in the CD7, and it's really understanding the options that you have here for automatic focusing and when is it better to use one option over the other. So I'm going to start with the most used and basic focusing routine, which is definite focus. Okay, so definite focus is going to use our hardware-based definite focus here, find surface action, where we ask the system to find the surface of our plate for us. So for example, if I were to click on this position here and I went live, I'm still on my transmitted light technique. Maybe let me show this view in a separate container so that we all have a better view. Okay. I can see here that I'm, maybe around the center of my organoid. If I click here on find surface, that again is going to call that hardware-based focusing routine that finds the surface of my plate. For these organoids, obviously they have a 3-dimensional aspect to it. So that surface is not going to be in the middle of my organoid, right? It's going to be at an offset. I can still use my definite focus to focus on them if they're all about the same size, for example. I can tell the system, okay, this might be the surface of my plate, but really I'm interested in this position. And I can just maybe scroll forward. Let's say this looks good. This is maybe around the middle of my organoid, right? And I can tell it, this is where my focus is, store that focus for me. When we do this, the system will now first find the surface of my plate and then measure the difference between my location right now and that surface. And it's going to remember that offset. That way, if I were to scroll away, I can always ask the system to recall focus. So it'll again find the surface for me and then apply that offset. So this is essentially what the definite focus routine would do. It would, for each position, apply that offset that you told the system to go to. So you see this cool organoid is having some like something is coming out of it. Let's press on recall focus. It'll again do that process where it would find the surface and then apply that offset. I could be in a completely different well, or I could be in a completely different area of my sample. So I'm just going to navigate somewhere else, maybe somewhere with cells as well, organoids as well. And maybe let me really focus out, right? So let's say that I really had no clue where my samples were. Again, the recall focus is using that hardware-based routine to find the surface and then apply that offset. And then that's where my image is going to get acquired. It would work also if I go into another well. So say that I go into another well, and I'm not sure if there's anything here. Oh, good. There's something in here as well. I could still use that same technique. And because from well to well, maybe the bottom of the well location might change, that offset tends to stay more constant. So this feature is very useful to help me focus on multiple wells without having to focus on all of them. So this technique would, when running the acquisition, and I'll run it on these two positions that we have for now, maybe I'll just capture just a quick oblique image of those two positions. When you're using definite focus as your focus strategy, when you start experiment, the CD7 will first tell you, it will ask you, okay, where do you want to initialize your definite focus? It's asking you, are you in your focal plane that you're interested right now? If so, continue the experiment, and then it'll measure that offset, remember that offset, and apply that offset to all positions, all tile regions that you're currently imaging. So you say, yes, I have focused. I know this is the location of my sample. I can continue the experiment. It'll stabilize the definite focus, measure that offset, and then go to the first position, apply that offset, take the picture. Go to the second position, apply that offset, take the picture. So this definite focus routine will automatically apply that offset to each position, each tile region. That way, I don't have to go in and verify my Z-plane for all of that, which, as you can imagine, here we're only doing 2 as an example. I imagine you were, again, measuring, say, 4 fields of view per well for your whole well. That's a lot of positions that you don't want to manually verify. You can use the definite focus and only focus once before you start your experiment. So again, the most important thing here is that you want to go live somewhere. It doesn't have to be in those positions that you're imaging, it could be anywhere else. And you can critically focus, find exactly where you want to take your picture and tell the system, okay, this is my Z-plane, apply that Z-plane to all positions. Is that clear? Do you have any questions about what the definite focus is or how it works? No question here is a bad question. Again, like I mentioned, this is probably the part that I want to make sure that everyone really understands, because if you want to use the automation in the CD7, the focus strategies are important. Okay, so when is it the most useful to use this technique? It's going to be the most useful for, let's say, for example, a monolayer of cells growing on a well plate, right? There, the cells are going to be basically flat on the bottom of the plate, and that offsetting between our plate and the cells is going to be very constant. It's going to be very small. It's going to be the same for all well plates. So great for those 2D monolayer applications. If you have slides, definite focus could also work great for slides. It'll also sort of find where that cover slip is and then measure the difference in between the cover slip and where your sample is located. I would probably not recommend using definite focus for samples like this, 3D organoids, because the offset could be different for different size organoids, right? Because the shape of my organoids is different, the size of my organoids is different. Maybe the organoids are not at the bottom of my well, but maybe they're floating a little bit. Maybe we're in a
Hello.
hydrogen. Yes, is there a question? Maybe not. Okay, maybe I just heard something. Okay, so for these kind of 3D like structures, maybe depth of focus might not be the best routine because it wouldn't really be able to compensate for different sizes of organoids or organoids that are in different locations in Z. In those cases, maybe a software autofocus routine might be more useful. So you have the option here of using a software autofocus routine, and I'll show you how that works. Maybe I'll go to one of these locations as well. The software autofocus routine will actually now acquire a Z-stack at a given position and find within the Z-stack what is the sharpest place of focus. And there's a few ways of how we can configure it. We have an automatic setting here where we can do a full or a smart search of our Z-plane. We can determine the range. It can be an automatic range, or we could manually set the range as well. But essentially what this does is, if we go live on our sample, maybe I'll show you how that process works. If you click here on Find Focus, or AF, or Autofocus, this will run our autofocus routine, which, as you see here, is acquiring a Z-stack, and then based on that Z-stack, finding the sharpest place. Okay, let me try this. The software autofocus obviously will be a software-based, essentially. Oh, I have DAPI selected as my focus references, and I wasn't acquiring in DAPI. Let me switch to DAPI here. And by that, I mean switch to DAPI. Let's select DAPI. Okay, let's do that again. So now we have our DAPI here. Let's apply that software autofocus routine again. And you'll see the system basically going through the Z-stack, capturing those images. Oh, we didn't find a maximum. That's interesting. I feel like that's definitely a good enough range for this sample. If we didn't find a maximum, it usually means that the range was not big enough to find the best place of focus. However, because this is a three-dimensional sample, that might be a little bit different. Okay, there it worked. I just took a snap where it took us to. So the software autofocus will again just aim to find within the field of view where we are, what is the sharpest place of focus, and it will start capturing a Z-stack around that position, and then determine based on that Z-stack where is the best place to take the image, and that's where it returns to. So that was the software autofocus essentially scanning the entire section of that organoid and then returning to this sharpest place of focus. So that would be an alternative method where instead of using that definitive focus routine, you could just tell the system, okay, at each of these positions or at each top region, go and do the software autofocus routine. And you can have it either do that full search that you saw there. So it searched through this full 200 microns that I set my search for. You also have the option of doing a smart search instead, where instead of going through the full 200, it just starts in the center and goes in one direction until it finds the best place of focus and then stops. So this is a faster acquisition. You can see here they found this as my sharpest place of focus. So this usually helps cut down the time that this step takes. However, it can fail if your sample has multiple local maxima of sharpness. It might not find the absolute maxima of sharpness, just a local maxima instead. As a summary, again, the software autofocus is not a hardware-based method like it was in the definite focus. It will actually capture a Z-stack at each of these positions, do the software autofocus for you, so that if you have this selected as your strategy here, then when you start experiment, the system will go to the first position, run the software autofocus, and then based on that, determine where to take the picture, and then it'll go to the second position, again, do the software autofocus, and then take the picture. So this is the result from each of those two. Any questions about what software autofocus is and the difference between definite focus? Okay, again, this technique is most useful for samples that maybe have three-dimensionality to them. Maybe the location of the samples varies in Z from well to well. So maybe I have well cells embedded in hydrogels, or maybe I have non-adherent cells, right? Maybe in those locations, a software autofocus might be better to focus on those positions if I can't use the definite focus. But by default, if you can use definite focus, I always recommend to use this first. This is a much faster process.
Sorry, I have a question.
Yes.
If you're doing a tile scan and maybe your sample isn't even throughout, so you might have different focuses in your tile scan. For each tile, can you set a different focus? Or...
Yes. So okay. Let's say that we had brain sections on a slide. Those brain sections can be easily 20, 50 microns thick. They might have different contours, different focal planes in your entire tile region. So what you would do is create your tile region. Maybe let me deactivate my tile positions, and instead I'm going to draw a tile region. So let's say that we were going here under tile region, and I'm just going to set it up by drawing a contour. I'm going to select my contour option, and let's say that we were drawing our tissue section here. So that was our tissue section. We can use, let's say, our software autofocus routine to focus on each of those tiles. So if we select our software autofocus as our routine, if we leave the stabilization here in standard mode, the system will actually run the software autofocus at each one of these tiles. So it'll do the software autofocus 39 times. You can imagine that that would probably take too long, right? That might be a little bit unnecessary because, okay, the focus might change from tile to, a few tiles over, but from tile to tile next to it, it might actually be very similar. So in that case, maybe you might want to go into expert mode and tell the system, okay, I don't need to focus every single tile. Why don't you just run that software autofocus for me every third tile or every fourth tile? That way, I don't have to spend so much time in the software focus strategy, and I can maybe just image and only focus every 4 tiles. So here, if you start your experiment, you'll see that it'll go to the first tile, run the software autofocus, take the picture, then skip 3 tiles. Then at the 4th one, it'll do it again. Skip 3 tiles, do it again, skip 3 tiles, do it again. I'll also note here that this tile region, obviously it's taken without stitching initially. So that's why you see that sort of like mismatch here in between tiles. This is easily corrected with a quick post-processing step called stitching that will just stitch the images back together. Okay, so that would be if you don't want to or if you don't need to focus every single tile, but you can get away with focusing every n number of tiles, and you can specify that n. I've also done it where maybe my tile region is small, but I have multiple tile regions to scan. Then I can tell the system, okay, just do the focusing once in the center of each tile region, and that's it. And then it'll just acquire that same Z-plane for that entire tile region. So you have the option here of either focusing every tile region or every n tiles. Does that answer your question?
Yeah, thank you very much.
No problem. You also do have an even more advanced option where you can insert for a tile region over here. You can insert for a tile region support points. So you can say, okay, maybe I want to do a little bit better than every n equals 3 or every n equals 2. You can actually insert or select support points to sort of lay out your focusing around your tile region. It's typically a more advanced option, but you do have that option to set that up if you want to. This is the support point here, distribution strategy, where you can tell the system how to distribute the support points if you want to. So that would sort of give you maybe a slightly more well-distributed focus strategy, and then you could use that instead as your focusing strategy. Okay, you do have the option of doing the same thing that I just did to go into the expert mode. So let me now hide my tile and region and go back to my positions. You also have the option, if you had your positions, to tell the system how often to do your focusing. So maybe you have your positions, and you have maybe that array where you have 4 positions per well, and then you move on to the next well. If the positions are close enough, maybe you don't need to focus on every single position. Maybe you can do it just every 4 positions or something. So that way you would focus on the first one, take the other 3, and then move on to the next well. This applies both to the software autofocus and also the definite focus, because maybe you might want to use the definite focus, but also stagger how often you do it to save acquisition time, because it's unlikely that you'll need to use the definite focus on all 39 tiles of a 39 tile array. Okay, there's a couple more options here. This next one is also fairly useful, is the combined software autofocus and definite focus. And really the way that it makes sense to do this is to use the definite focus as the start of your software autofocus. This could be very useful for a sample like this, where we could tell the system, okay, first use the definite focus, find the bottom of the plate, and then use that as my start for my software autofocus, because like you saw here, I can have either a relative range or a fixed range for my search of my focus strategy. And if I'm using a relative range, it's always based around the specific location where I'm at. So if I just put in this plate and I don't do any focusing at all, this software autofocus might fail if it's not within 200 microns of my sample. So I can always tell the system, okay, first use the definite focus, take me to the bottom of the plate, and then run a software autofocus routine around that bottom of the plate. That's what this would do. And the last one is to use the focus surface or Z values defined at tile setup. So if you were using that support point strategy that I showed you for your tile region, this is what you will want to use, because then this focusing routine, focus strategy would use that setup that you made for your support points to capture the tile region. Or you could just say, okay, I'm manually creating this focus values and I'm focusing on each position to tell it exactly where I want to image. So maybe what you might want to do is you might go live and you might, see, okay, this is exactly where I want to be, or maybe I want to focus a little bit more. So you might, change it and you're like, okay, this is the position I want to image. Don't change it. Don't do anything automatically. You could just tell it, this is exactly where my position is. So you could create your position at this place, or if you've already created your position, you can adapt the Z value and you can tell it, okay, this is position 1. This is where I want to image. So you can change the Z value. And if you change the Z value here and you have your option, your focusing strategy here to just use the tile setup, then the system will just go to that defined Z value that you created. The one thing that could be useful here is if you have samples that are really varied in or heterogeneous in Z. So even more than this, I've only used this very rarely. The one example that I had was this plant leaflets that were cutouts from plants, and they were basically just dispersed in 96 well plates. And the Z position between each of them varied so much that the software autofocus wasn't working, the depth of focus obviously wouldn't work. So what I ended up doing was creating positions and then verifying the positions so that I could tell it, okay, go to each of these positions, and you could even go live. And then you can click on that position, and then you can tell it, okay, run autofocus for me. And then you can visually verify in front of you, okay, run the autofocus and tell me what that looks like. And then you can say, okay, that looks good. And if you're happy with it, then you can then move on to the next point. So this is sort of a last resort. If nothing automated works, you could still use this verify position workflow to manually verify your positions before starting your acquisition.
So I wanted to say that there's a couple of different automated high throughput microscopes on campus here that I run. But this is really great in terms of if you do have a difficult sample, you can go to these links to get it focused. So I think that's really going to be interesting. And the other thing is there's some analysis that we can't do on these microscopes that are the automated ones that are really meant for high throughput. So we have in the chat, maybe you've seen it, Rocio's kind of request, which might help guide what you take in the confocal mode for us to move to the analysis.
Sure. Let me take a look at the chat here.
But this is very nice.
Okay, I think I saw this. Orwell's analysis. Okay, so try to get some analysis done. Okay, some localization of cell expression within the organoid. Okay.
And distance from the center.
Okay. In 3D, that is a little more challenging, obviously, because we're not looking at distances in 2D anymore, but we sort of have to factor a 3D distance. Obviously, for that, we'll have to go into confocal acquisition. And we'll have to play around with things there a little bit. Typically, when you go into 3D analysis, you're talking about maybe going into Airys, which is our partner 3D analysis solution to make those kind of measurements. But I can show you some of the basics that we can do within Zen as well.
Okay.
Okay, so this was where was I? Showing, okay, if you really had to go to the absolute, more involved way of focusing for your complex sample, there is a way of setting up your positions and verifying all your positions, or even building multiple offsets of your positions. So you could maybe, if you wanted to acquire a time lapse and all of the positions were different, you could tell it, set a definite focus, particular offset for all positions, and then use that offset throughout your entire time lapse. Those are definitely more advanced setups, but they're definitely doable within your focus strategy. Okay, so that was Z-stack styles. Time series, this will now be sort of an easier setup now that we've gone through everything else so far. Time series will enable this time series window that will let you choose if you want to image, say, continuously for 10 cycles. So if you were taking like a calcium imaging application and you needed to image, so many frames or so many cycles, you could set up your cycles here. Or you could tell it, okay, image continuously for 1 minute. So again, if you had that calcium experiment or something else where you wanted to image continuously for 1 minute, you just choose here your duration and your interval will be 0, right? You're basically just imaging as fast as you can. You could also do it so you're doing like a long-term time-lapse imaging. So let's say, okay, I actually want to image for 12 hours. So you can select your 12 hours. And then obviously you wouldn't image continuously because that would be an impossible data set. But you could image, let's say, every 15 minutes. So then you could say, okay, every 15 minutes for 12 hours. So that would be your time-lapse experiment. If you're doing a time-lapse, then I do recommend that you also activate or set up a focus strategy. Specifically, if you're doing long-term live cell imaging and you have heating involved, you can definitely use the definite focus. So for example, if you have your 2D cell culture and you wanted to see those cells maybe migrating or moving or dividing, right, in 2D, then definite focus would be the best way to make sure that you're in the same focal plane at every single one of these time points every 15 minutes for 12 hours. That way there's no thermal drift. All of these options can be combined all together. So again, let's say that you had, let's say, you could have maybe cells that were growing, maybe not just in a monolayer, but maybe in a multi-layer. So maybe you might need a Z-stack. And then you wanted to image 4 positions per well. Then you would have to activate all of your multidimensional acquisition components. And then you would just go by one and set up what each component is going to look like. So for your positions, you might say, okay, I want to image here under your positions or your tiles. You can set up how many positions you want. Then you would go and set up your Z-stack. So maybe you might want to go into those positions and set up your first and last. You could also do it. Typically what I do is when I have multiple positions is instead of using a first and last, I use a center because I know that I'm going to be using a focus strategy. So it makes more sense in my head to use a center distribution. And in that way, typically I do start with the first and last, but then I use that just basically to determine my range. So let's say, okay, my Z-stack, I want it to be 60 microns. So then you would just go here under sensor, right, center, right in your 60 micron range. And then your software autofocus or definite focus routine would take you to the center of that range. And then I would acquire that range around it. So that would incorporate both a Z-stack with your positions and with time lapse. So you could do that Z-stack acquisition, multiple positions over time, and it would be centered around that range. And again, your focusing routine, whether it's definite focus or software autofocus or combination of both, would take you to the center of that Z-stack, and then you would capture the range around that. You could always apply an offset as well. So if your sample was heterogeneous and maybe the software autofocus or the definite focus was giving you a slightly lower or higher plane than what your Z-stack you want it to be, you could always apply an offset to that Z-stack based on that center. Okay, so that covers multidimensional acquisition in the CD7, most of the automation routines, most of the focusing routines. There are a few other options here that you can activate. If you're doing a time lapse or anything that's more involved, I recommend that you just activate the autosave function, and that will make sure that the data gets written and saved as it's acquired, and you can create your folder with your name, with the prefix, with whatever ID you want. That way, if something happens, you're gonna make sure that that data gets auto-saved for you. And you do have a few other options in here that maybe I might leave for a later time, since they are more advanced options. And at this point, I would want to go into now the setup of confocal imaging to make sure that we cover that as well. Does that sound okay?
That sounds great.
All right, so this is going to be more simplified. I'm not going to do time series. I'm not going to do tiles. I will leave the Z-stack activated here as obviously confocal, makes more sense when we think about that with Z-stacking. The last thing I'll mention here before I move on to that is everything that you build so far in here is will be comprised, or everything is going to be contained within this experimental workflow. But you do want to make sure that you save it, because if you didn't do anything, if you didn't save it, your setup will basically be gone. You could use it, right? You have it available. Actually, next time if you pull that image up, you might be able to extract the workflow, but you would want to make sure that you save it if you want to use it again. So if you're building a workflow that you're going to be using often with your positions, with your Z-stack, and with your focusing routine, you only need to build it once, and then you could keep calling it whenever you're ready. The one thing that I will note that is the biggest thing that is not saved in a workflow unless you go in and bake it into the workflow is your acquisition magnification. Because as you've seen, I can easily change between magnifications and my setup will adapt to that magnification, right? If you want to always make a workflow that always images at a specific magnification, you do want to bake that into your imaging setup, which is available right here. And you can tell the system, okay, I want to only exclusively use this workflow to image at this 10x combination, for example. So in that case, you would bake in, including this setting, my 0.5x magnification changer and your objective right here. You could even bake in the correction ring adjustment if you're always going to be using this for the same sample. When you do this, then this workflow will always call this magnification to image. That way, if this is exclusively what you're doing, I would probably even recommend saving it in the main, because since it's now baked in here, if I go live, it's happy. It's at its 10x magnification that I'm going to be using. But if I were to move to a different combination, let's just exaggerate this and go to 40x. So now I'm at 40x. If I go live, you'll see that it changed to the 10x for me. So that's because I baked it into the imaging setup, where this channel that I'm using and this whole setup will exclusively be using this combination. So that's just something that I like to point out, because you might want to build a fully automated workflow where you just put in your sample and click on start experiment and walk away. But if you want to do that, then you do need to make sure that you're calling the right objective in your setup. Does that make sense? Any question about that? I'm going to revert this back to no objective calling, since typically I like to put in the plate and verify myself the magnification that I want to use before starting the experiment. Okay, so let's create now a new workflow for confocal imaging. So I'm going to label this LSM, organoid 24. Okay, and I think most of you are actually pretty familiar with confocal imaging, so I'm actually not going to go into too much detail here, because this will actually be like almost exactly the same as your other confocals, at least the ones that run in Zenblue. So I'm going to just do a smart setup here. And I have these Widefield channels that I have built previously. I'm just going to get rid of them and go back into my LSM mode. And here I have the option of choosing the confocal mode that I want to use. So in this version of the LSM 900, you have the LSM confocal. So this is purely traditional pinhole-based confocal. And you do have 3 detectors in your system, so you can configure up to 3 different tracks. Or you also have the Airyscan detector, which is that 32 array detector that can go into push the boundary into super resolution beyond the traditional confocal by using that honeycomb array to gather your signal. And it does have 2 modalities on how it can be used. We have HS, high sensitivity, which can give you the highest level of resolution, up to 1.5 times the resolution compared to traditional confocal. Or you have the multiplex HS, which has a slight compromise in resolution, up to 1.4 times the resolution of traditional confocal, so just a little bit less than HS. However, it can capture 8 to 16 times faster than traditional confocal, while HS can only capture, I believe, is 4 to 8 times. So multiplex gains a lot of speed in capturing with only a slight compromise in resolution. So for large 3D samples, I typically just use multiplex. Like for organoids, I would just go to multiplex mode because it would dramatically increase my speed and sensitivity and signal to noise ratio so that I could capture those bigger samples faster. If my sample was small, so for example, just 2D cells, then maybe I don't care so much about the speed because it's going to be a small Z-stack anyway. So the HS mode might have an edge because I could maybe get a little bit more resolution, right? Up to 1.5 times confocal. Let's start with just a basic confocal track. And I'm gonna let's just do maybe these 3, 488 and 647. When I add those 3 channels, you'll notice here that I have a few options of how that could be set up. Fastest, best signal, or smartest. I almost always just go with the smartest. For most users, that's going to be the easiest way to set up, and it'll be a good compromise in terms of signal and speed. However, you do have some other options where you can choose your speed. So if you want to have the fastest speed or the best signal, obviously the best signal here is going to use our 3 different detectors, and you'll configure the channels going to each detector appropriately to give us the best signal. So you have the option here of what you can choose, and you can also have a representation here of what each track is doing. And maybe if you're okay with, here this fast, it might actually be, pretty good. It has a good speed and a great signal, so maybe that could be the case. But you do have the option here of what to choose, and then once you hit OK, it'll create those tracks for you. And then you would do your traditional setup. If you use confocal before, again, I'm not going to go into too much detail here, since you have some very experienced users, probably more experienced than I am, that can also go over you go over this with you. But essentially, you can set up for your confocal tracks. If you want to scan by frame or by line, you can have presets on your pixels. You also can set up your bidirectional acquisition. Maybe you might want to do some averaging. You can choose all those options here. You can also adjust your scan speed. So maybe you might want to slow down the scan to get a little bit more pixel dwell time to increase your signal to noise ratio. And then for each channel, you can obviously see your tracks. You'll be set up an acquisition that has within one track 2 different channels, and then 1 second track for DAPI. So for each of these, you could set up your laser powers. You can set up your pinhole, which I typically do with the longest wavelength. So I would go here with 647 and set that up to my 1 Airy unit. And then I would sort of just match the other ones to have the same section, so 3.4. And then I would just make sure that all of them have that 3.4 section. And then beyond this, then it would be a matter of setting up your gain. I typically start with like around 700 to 750 gain for my channels, and then I just play around with my laser power a little bit during that setup. Again, there's a lot more that can go into here. I'm not going to go into too much detail here in the interest of time. If you have any questions about how to optimize these confocal setups, please let me know. Otherwise, I might just switch here and show you a little bit of the Airyscan, since I think this might be where you might find some differences compared to your other systems. If I wanted to do an Airyscan acquisition, I always just go back to my smart setup, and I can just select here my detection mode, and let's add our multiplex track, since that's what I would recommend for these larger 3D samples. And again, I usually just go with the smartest recommendation here. Okay, with the Airyscan, you do want to check that you have an optimal configuration, and it'll actually tell you if it's optimally configured or not. I'm guessing here, the first thing it's going to tell me is that I need to crop my field of view. Typically, the Airyscan recommends a zoom that's at least 1.3. It's very easy to adjust. You can just go here under your crop area and increase that to 1.3. And that's usually sufficient for that. And then here we have our frame size. So we might either adjust our zoom factor, or we might select one of the 2 different options of how to select my pixels. This is going to be specific to the CD7, these 2 options. So maybe I'll spend just a little bit of time explaining them. Again, multiplex mode has that possibility of increasing the speed 8 to 16 times the speed of a regular confocal. And that will be a setup here using this HS2Y mode. The way that this is done, and it can get fairly complex here. If you have some questions, you can always use our help menu to open the help menu specifically for whatever you might have a question on. In this case, for the Airyscan acquisition, and I can give you more details into the parameters here that you can set up for your LSM tracks, including the options for your LSM setups or your Airyscan setups. And it has a pretty good description. It's very well indexed. So I do recommend that you have some questions. If you're unsure about what something means, really make use of this help menu because it'll really show you more details here. But what I was going with this difference is just the way that the 32-channel detector is sampled. In one of these ways, it just takes 1 pixel that's a little bit bigger, And in the other case, it makes actually 2 pixels. It can fit 2 pixels in the array, so it can scan a little bit faster. I can maybe describe this in more detail if you want me to. It does get a little bit technical, but for now, what I would say is I typically recommend using this HS to Y mode, which does achieve better signal to noise ratio. Okay. So let's set up our Airyscan here. I typically will always, just use DAPI as my initial setup, and we can go live in here. Let's increase here our laser power. As opposed to Confocal, typically when running in Airyscan mode, I recommend using a gain of 800 to 850. That's my recommendation. Let's center here on one of these organoids. Okay. And typically, I'm going to adjust my lasers either with an 850 or 800 gain. And I am aiming here to fill quite a bit of my dynamic range of my detector, which you can see over here. Or if you're used to using the range indicator, you can also use the range indicator if you wish. One thing that you'll notice here versus other Airyscan configurations is the system automatically aligned the detector for me. It does this whenever I'm in live mode. And it's this option here that says adjust in live mode or in continuous scans. That way I don't have to do the alignment myself. However, if I do see that the system is maybe struggling with doing the alignment automatically, I could always stop my live view. And under the Airyscan icon here, I could deselect that option and then manually adjust my detector if I need to. You can see here that it's already aligned, so I don't really need to make any changes. This is what I want to see, my brightest signal right in the center of my 32-channel array. Okay, I'm just gonna revert this back to the default of automatic alignment, since for most users, that's gonna be the easiest setup. So like you saw, I only had to really just play around with my LED power, laser power output, because now we're using our lasers for illumination. And then once I'm happy with my laser for that channel, I would just then select the next channel. And do the same thing, maybe increase my laser power a little bit. Probably already pretty close to being good. Could try the 647 channel as well. Increase our laser power. And I'll let you decide if for your samples, an 850 or 800 voltage might be the best. That is something that I don't typically change too much when using the Airyscan detector. I typically just select here the bidirectional, and actually for Airyscan, averaging usually doesn't make much difference, if any. So for the most part, I don't typically do any averaging on the Airyscan. I've seen some users maybe do 2 times averaging, but beyond that, there's definitely no added benefit of doing more averaging when using the Airyscan, because essentially, because also of that 32-channel array, we're already averaging essentially. So I took a quick 2D Airyscan, so no Z-stack yet, just a quick 2D of that one single plane where I'm at. And then in this 2D view, you'll see here the raw data. And because this is an Airyscan acquisition, you do want to make sure that you process the data with the Airyscan processing. And you can do that right off the bat right here in this Airyscan tab. And you can select your current image and create the processed image. And that's going to be your actual final result of your Airyscan processed data. Okay, maybe let's set up just a quick Z-stack so you see how that process works with the Z-stack. So I'm going to activate.
I wanted to ask why you dropped out the other channel that was the 560. What was it?
568.
Yeah, 568. Was that just to simplify things? Because I think that might be an important one.
Yeah, sure. It was just to simplify things. I didn't see a bright expression with it in Widefield, and just to, the setup would essentially be the same. You would just add that channel, but just for the purpose of today's
thinking
training.
to have the data set to play with. Rocio, maybe it's not an important one. You can put in the chat.
Sure, yeah, and I mean, obviously, for the purpose of today, just to make sure that everyone is familiar with the setup of Airyscan and with the options here, and just to simplify things with just a few channels. Because obviously, adding more channels, if you're using the Airyscan, we would add more time to the acquisition, because if you're just using confocal mode, you could use all 3 detectors. If you're using the Airyscan mode, there's only one Airyscan detector. So all of these need to be imaged sort of sequentially as opposed to at the same time.
I see. So there's no issue with the crosstalk or anything from that.
That would be obviously dependent on how you set up your smart setup. So that's why, when you do this smart setup, it'll show you if there's potential of crosstalk. And if we added a 4th channel, it would add another channel here and it would give more options. And it might actually warn us that 568 might be close to 647. So we might make a recommendation on how to avoid the crosstalk with that channel.
Okay. Yeah, Rocio said that was an important one, but for the demo, we don't have to. Maybe we can get it some other time.
Sure. Yeah, definitely. Yeah, I'm just trying to be mindful of the time because we only have about 20 minutes left. But let me show you now, let's just set up a quick Z-stack here with our Airyscan. So I'm going to go live here. And let me select maybe just my DAPI just to get my bearings around my sample. And I'm going to scroll here to my Z-stack view. And maybe I'm just going to capture a sliver of the organoid just for demonstration purposes. So I'm scrolling using my scroll wheel on the mouse again, keeping that control key pressed on the keyboard to scroll back. I'm just going to set this here as my first. And then I'm not going to capture the whole thing. I'll just capture a few slices again, just in the interest of time. Let's just capture that and see how big that is. Okay. So we're going to press here on optimal, 1.3. Let's go back to the center here. Let's see. Oh, the frame size is not set up for optimal capturing. Let me take a look here. I'm already at 1.3. Maybe I might need to go a little bit. Let's see. Okay, that looks fine. One thing that I will say is obviously when you're in confocal mode or in Airyscan mode, cropping your region will always increase your speed of acquisition, right? Because we're not spending time capturing, other area around it. So do use the crop function often and try to get as close to your sample as you can. That way you can get away with a faster capture. Okay, so I set up my acquisition here. I actually don't even remember if I set up the Airyscan Acquisition to capture. It's doing all, it's capturing by line. So it's switching tracks every line. We could also set it up so it switches track either every frame or for large Z-stacks. What I actually usually end up doing is I tell it to do the whole capture of each track for the whole Z-stack and then move on to the next track, which can be useful, particularly also if you're changing your filter in front of the detector, which is very useful if you're having problems with crosstalk or bleed. But for now, we'll just leave this line acquisition go through. Maybe I'll just show you once this is done, what the other different options here for track switching. Do you have any other questions here in the meantime while we wait for this, minute and a half? Okay.
I've lost the picture.
You mean the live view?
Yeah,
Um,
maybe it's just my phone.
Did anybody else lose live view?
I can see it.
No, I can see.
Okay. All right, so we'll just wait for this to go through. Again, we're switching here every line, which is why we can see all of the colors sort of acquired almost at the same time. But I'll show you some of the different options that are available as well. Okay, so that was our acquisition. And again, because this is an Airyscan file, we do want to process it. And in this case, this is a 3D file. So the best processing is the 3D. And then you just click here on create processed images, and it'll do that 3D processing for us. You can see the progress here under the status bar.
So Sebastian, if people are acquiring a lot of Airyscan data, then it'd be a lot better to send it to the processing station, right?
Yeah, for sure. So then you could make use of the direct processing to send the data over there. And that way, maybe you can, just send the data over there, keep running your acquisition, and then you can, not burden your acquisition computer with these processes. So you can keep on acquiring and on parallel do your processing. Okay, once you have your processed image, then you can do whatever you need with it. We can create this 3D display view here. It'll take just a couple of seconds to render, but then we can scroll in and out. We can also adjust our display here. So this might be a little bit bright. So I'm just gonna maybe decrease my display adjustment just so I can actually see things a little bit easier. And then you can always rotate your shape around again. We only capture a little sliver of it. But you have these options here. You can always also just go back to your 2D view, and maybe you might create an extended depth of focus, like a maximum projection, to just sort of see the whole thing together. So you might come here and then run your EDF extended depth of focus and create your maximum projection. Okay. Let me show you just real quick some of the other options for your tracks. Again, we were switching here every line. If your Z-stack is quite large, it might be faster to actually run the Z-stack, or maybe by frame or frame fast, just so you can capture a little bit faster. It could also be useful. See here that we have a filter running for our detector. I don't think we are right now changing that filter between channels. We're just sort of capturing it all and then just using the laser excitation to distinguish our channels. But if you have some channels that are problematic, you might want to maybe, cut your actual capture of emission. And if you do this, then switching every line would be very slow because you have to change that movement a lot. So if you're doing this, then maybe doing the Z-stack switching might be the better way of doing that. Okay, so this essentially covers everything that I would want to cover for our first round of training. The goal of today, again, was to get you familiarized with the system, get to explain the different options for multi-dimensional acquisition, Z-stacking, tiling, adding time lapses, how to set up channels for Widefield, how to set up channels for Confocal and Airyscan, and also how to optimize some of your imaging parameters, including your focusing strategies, and what each of these mean and the best time to use each of them. I'll let you guys, play around with the system for a little bit, and we can always schedule a follow-up session to maybe look at some other workflows in more detail, or if you need assistance with particular workflows as well.
Thank you, Sebastian. That was awesome.
Yeah, thank you. I have one question. If you want to do kind of for speed, like a combined wide field, like maybe 2 colors in wide field and 1 in confocal, or Airyscan, that's possible, right?
It is possible. It'll not work with every mode of confocal. And my memory is not going to fail me on which is the method that it's not suitable for, but you'll know right away, because if you add, a couple of confocal trials, like let's say that we didn't need super resolution for DAPI, right? Just a wide field picture is suitable. So maybe we could do our DAPI in wide fields, and then we could do the other channels in our confocal or Airyscan. It'll tell you once you try to create it if it's suitable, if this particular combination is suitable or not. And I want to say with confocal, everything works. I think with Airyscan, yeah, I just selected confocal. So that means that we can do this for confocal. And I think for Airyscan, it'll warn us, it'll tell us that it can't do, I forget if it's multiplex or HS, the one that is not suitable. I want to say it's multiplex, the one that doesn't work with Widefield.
Okay.
And then what you would do here is just set up your channels in the same way as I did before. So just start with your DAPI, set up your camera and your LED power setting, go into confocal, set up those settings, set up your Z-stacking. One of the great benefits that you get from doing this modality is now you can use your Widefield track if you want to do something like software autofocus. That can be super useful, because that would obviously be harder to do with a confocal track because it would take longer. But now you can just use your Widefield confocal track, so you can select your software autofocus to work with DAPI as your focus reference. And then you can capture your Widefield DAPI along with your confocal tracks on top of each other.
Cool. Okay. Yeah. Yeah. We'll obviously have to try out the system, and we'll probably have 5 million questions soon.
Yes, for sure.
Yeah. Does anyone have burning questions?
Okay.
right.
Okay, so definitely go ahead and try it out. Don't be intimidated. You won't break the CD7. Like I mentioned, it has a lot of safety features built in. So just go ahead and have fun. Try different things, try different tracks, try different combinations. This system is very powerful and also very versatile. So there's a lot of things that you can do with it.
Okay.
Thank you very much.
Thanks so much, Sebastian. Really appreciate it. And we're definitely looking forward to putting it to use.
Definitely. Okay, awesome. If anything comes up, just let me know. Otherwise, have a great day, everyone.
Okay, thanks.
Bye.
Thank you.